This method makes super-resolution cryoimaging possible on whole biological cells to precisely identify cellular structures. It can also be used in conjunction with other modalities as part of a correlative imaging workflow. The main advantages of this technique are that super-resolution imaging can be rapidly done in cryogenic conditions using conventional fluorophores and relatively low light doses.
CryoSim is a powerful tool that provides insight towards understanding cellular ultrastructure dynamics in response to internal or external cues. Its application include vaccine production and rollout, quality control, post-market surveillance, antibody engineering and optimization, as well as nanoparticle characterization.
Maneuvering samples within the cryostage takes practice to ensure the safety of the grid. It's also best to familiarize yourself with the controls in the cockpit window before data collection.
To begin, remove the lid from the external dewar of the cryostage and pour filtered liquid nitrogen until it's approximately a quarter full. Wait until initial boiling subsides before pouring more and fill the vessel to about 2/3 full. Replace the lid carefully, pointing the nozzle away from the handler as the liquid nitrogen boils out. Once the liquid nitrogen has stopped coming out of the outlet, place the outlet pipe over the stage dewar on the cryostage. Plug in the power source, connect the USB cable, and plug in the external dewar to the stage. After delivery of liquid nitrogen, press the release button on the cryostage to allow it to enter the sample chamber, and wait